Hi all, welcome back. And for this set of lectures, we're going to talk about the action of the heart. So we're going to go through kind of what controls the heart, what maintains its rhythm, etc. And we're going to talk a little bit about pressure and how that's related. Okay, so first off, when we're talking about heart actions, like we talked about a little bit before, heart is a muscle, and so just like any other muscle, you're going to have a contraction where actin, myosin heads are linked up and fibers are moved, okay? And then you're going to have a relaxation where the linkage is broke and the fibers can go back to their original placement. Contraction is called a systole or systolic, and the relaxation is called diastole or diastolic. Okay, and so depending on what chamber is contracting or which chamber is relaxing, then that'll get the term that we're talking about. So if we're talking about a cardiac cycle, okay, so this is what's occurring during a heartbeat. First, the atrium, the right and left, will contract. So we refer to that as the atrial systole. Okay? And while that's going on, the ventricles are relaxing. So we call that the ventricular systole. Okay? Then when the ventricles contract or go through systolic, okay, the atrials relax and become diastolic. Okay? And then at the very end of the heartbeat, both chambers will relax. They'll both be in diastolic positioning. Okay, so we've talked about many things when it comes to like the cardiovascular system so far, but part of the reason why we touch on these things is because we want to know how modern medicine might allow for fixes or deal with complications when it comes to something like the heart. Okay, so real quick, I just want to briefly touch on some things that are going on in science when it comes to the heart. And of course, heart transplants are maybe not super common, but they're becoming more common as time goes by, and a donor heart is needed for this. Right now, the donor needs to match your blood type or at least be an O type heart. And so that kind of limits the heart transplant actions. Now, most recently, a couple months ago, a team of scientists in Canada were able to remove the enzymes that basically they are able to use enzymes to remove the sugars. So remember when we were talking about blood typing, that is an antigen that occurs on the outside of the blood cell. Well, it also occurs on the outside of organs. And so they're able to use enzymes to remove those. So it looked like the organ that they're transplanting is an O-type organ and has no antigens on the outside. If we're able to do this, then it will not matter what blood type the donor heart is if we're able to enzymatically remove. And so we're in the trial stages of doing so. And so it looks like some very promising research right now. So heart transplant is that's the goal, okay? But because of that waiting time, etc., other things have to be developed, like left ventricle assist devices. Normally, the left ventricle, because of its prevalence and because of its role in the heart, in that it's forcing the blood to the body, it plays a major role in the heart, and often it's the piece of the heart that we really, really need to be working at its best. And so sometimes we'll put a Lavid, which is kind of a half heart, just a diversion pump. Okay. Again, these are not permanent. These are just hanging around till that donor heart is found. Other things, titanium, plastic, artificial hearts. Again, these are put in till the donor heart is around, so they don't have a very long life. But sometimes it's the patient's elderly, and you want to give them a little bit of time, so maybe their family can show up and say their goodbyes, etc. So you might put in a plastic artificial heart, knowing that they're probably not likely going to get a heart from a donor, and this will give enough time for the individual's families to say goodbye. Okay, another great avenue of research, not just with transplants, but often what's happening is patches of cells are dying on the heart when someone has a heart attack. Part of the heart muscle is dying, it's decaying, and so modern research is using stem cells to make what we call heart patches. So if you know the region of the heart muscle that's dead, it is possible, or at least hypothetically possible, to remove that dead decaying material and put stem cell heart patches in there. And as long as the heart muscle, the stem cells differentiate into heart muscle, okay, and you get these bridges using the normal cell material, then you can get the transfer of the action potential to go all the way across the muscle. And this is shown, mixed reviews on this technology, but it's getting much better. And so hopefully in the near future, we'll be able to treat hearts with stem cell heart patches. Okay, and then of course there's been technology that's been around for a very long time. If a heart valve is faulty or damaged, so tricuspid, bicuspid, one of the semilunar valves, we've been using pig heart valves for a very long time. The problem with that often is, again, they don't last that long, so you'll have to have a replacement heart valve. But nonetheless, a heart valve from a pig, 5 to 10 years kind of lifespan, and then you might have to get that one replaced. It depends on the technology, our ability to remove the antigens that are occurring on that heart valve so the immune system doesn't attack it. And so right now we're making pigs that lack those antigens. So we're breeding pigs that we've removed the antigens so their heart doesn't have the antigens, and then we can use their heart valves in human transplant or human repair cases. Okay? So lots of avenue from a technology perspective to try to keep this system rolling. Hey, again, another really important role of when you're using like stem cell heart patches is the intercalated discs. Those need to line up, and so when you put a patch in, you need to have these intercalated discs reform, and that will allow for that action potential to move from one cell to the next. Okay, and we talked about cardiac muscle lots of lectures ago, and its role, kind of how it's different than skeletal muscle. Yes, it's elongated, but it has a single nuclei per cell, and it's not elongated to the degree that skeletal muscle is, but part of that transition from that action potential moving from one cell to the next really has to do with these intercalated discs. It also has to do with these channels, the Desmones channel and the gap junctions. Okay, they allow for things like sodium, potassium, calcium to move freely and allow for those action potentials to continue to move down. This is very important from a heart perspective, because if the SA node, which we'll get to here in a second, if it sends an electrical pulse, you want all the muscle fibers kind of firing in unison, so you get one beat of the heart, and you don't have, an offbeat, or you don't have one patch firing and the other patch is not. And then that'll cause an arrhythmia, and the heart may malfunction. And we'll talk about this here in a second. Okay, so when we're talking about that conduction system, the ability for a electrical impulse or an action potential to be started and then distribute that action potential across all the myocardium, across all the cells, is really that cardiac conduction system. And it starts with the pacemaker, or the SA node. Okay, the sinoatrial node, that is the pacemaker of the heart. It's what keeps the rhythm to the heart. Okay. The importance with this is it's normally keeping a rhythm of about 100 beats per minute. Now, if your heart rate is less than that, well, the interesting and the nice thing is the SA node is controlled by the autonomic nervous system, which is controlled by the both fight or flight response, okay? And it's controlled by the parasympathetic part of the nervous system, which is rest and relax. So the sympathetic nervous system is fight or flight, okay, and often maybe norepinephrine is being released, and that will increase the heart rate. So maybe the SA node is keeping it at about 100 beats per minute, but you maybe are afraid of snakes or something, and you see a snake, and now you get this big rush of adrenaline or norepinephrine, and your heart starts beating much faster. The opposite is true. Some individuals who have a much lower heart rate, maybe you're not at that 100 beats per minute, hey, maybe you're sitting at 60, 70 beats per minute. Okay, it means that often you're in the rest and relax state, or the parasympathetic nervous system via the vagus nerve is stimulating the heart to slow down. Okay, so it's enticing the heart to slow its beat down, to slow the sending of the electrical pulse down. Okay, and so a lot of times athletes who have trained themselves to up their heart rate when they're exercising, when they go back to non-exercising, their heart rate will be much lower, okay, because it's being stimulated from the vagus nerve to lower the heart rate, because they don't need to pump the blood as fast as what they do when they exercise. Okay, now, so that electrical impulse sent by the SA node, okay, it goes to internodal arterial muscles, so it goes through the atrium, both right and left atria, okay, is stimulated. Okay. Along with that, that impulse is picked up by junction fibers, and that's sending the impulse to the AV node. Okay, so the atria can fire, so the top portion of the heart, both left and right, they're contracting, okay, but that impulse, that electrical impulse, that action potential doesn't move from the atria to the ventricles directly. It has to move through junction fibers to the AV node, or the atrioventricular node. Right? So the atrioventricular node, it's what moves the action potential from the atrium to the ventricle. And it does so through the bundles of His. Okay? And so there is a little bit of a delay between the atria contracting and the ventricles contracting. Okay? And that kind of what's associated with this is that transfer of the action potential from the SA to the AV, from the AV to the AV bundles of His, right? And then that will move that electrical impulse to the ventricles. Okay, it moves the electrical impulse to the ventricles through the left and the right bundle branches. Okay, so this is traveling to the right side of the heart, the bundle. Okay, travel the left would go to the left side of the heart, and that would stimulate the heart muscles in those regions to contract. Okay, the fibers that run through the heart from the bundle branches are Purkinje fibers or Purkinje fibers, and that's what fires the myocardium or allows for the myocardium to contract, to carry out the actin-myosin interaction and contract the heart. So here you can just see kind of what I covered, and we'll just kind of go through it real quick in visualization. So the SA node, the pacemaker of the heart, it's going to beat at 100 beats per minute most of the time. Okay, it depends on the SA node, it depends on the individual, but this has a autonomic response. It's just controlled to beat at that rhythm. So it's going to send those electrical pulses at that rate. Okay? When it does, it'll move through the muscle fibers across the interarterial septum. Okay, so it can move across the septum, so both left atria and right atria can be contracting at nearly the same time. Okay? That electrical impulse will move through the junction fibers into the AV node. When it's in the AV node, it then moves into the AV bundle. It's carried by the bundle branch, okay, the right to the right side, the left to the left side. Purkinje fibers then take it up and move it all the way around throughout all the myocardium, okay, including the papillary muscles, which are going to control your valves. Okay, your bicuspid and tricuspid valves. Okay, and then you get that ventricular stimulation, so it will contract. All right, let's look at ECGs or EKGs, depending on what country you're in, but an electrocardiogram. Okay, we talked about these before. We talked about them in lab. Okay, it's just measuring that electrical impulse that is being generated from the SA node to the AV node. Okay, so it's really just a measurement of the electrical impulse that's occurring. So you're going to get an electrical impulse that happens a little bit before a contraction. So the P wave is the atrial depolarization. Okay, so if you see a P wave on an electrical cardiogram, okay, right after that P wave starts, you'll start to have atrial contraction. Okay, the QRS complex. Okay, you got 3 waves. You're going to have ventricular depolarization. You're going to have atrial repolarization, but it's kind of hidden in the QRS complex. And then after the QRS complex, or after that ventricular depolarization, you're going to have a T wave, which is ventricular repolarization. Okay? And again, these are waves of energy. That's what the electrocardiogram is picking up, is the action potential that's occurring. And it's in conjunction with the contraction or relaxation of the muscle. Okay? And so we'll kind of cover these here. So here's an ECG or EKG pattern. So you get this initial SA impulse. Okay, so you get this initial little bleep in millivolts. Okay, after that starts, then you get the initial contraction in both chambers. Okay, so it starts off in the right atria, moves across the septum into the left atria, and then you can get both chambers starting to contract. So by the end of the P wave, the electrical impulse has moved across or throughout the entire atrium. And it's now from the right atria, it's now at the AV bundle or the AV node. Okay. And so this is known as a P wave. That first little bleep, it's a P wave, and that coincides with the contraction of the atrium. Okay, so it's contracted. Now that signal moved from the AV node to the AV bundle, down the bundle branches, okay, both left and right, okay, and then that signal starts moving back across, okay, the myocardium of the ventricles. Okay. When it does so, that's where you kind of get that QRS. Okay, and here you can see the QRS finish. That means the electrical impulse has made it through all the myocardium of the ventricles. Okay, in the kind of the back side of that is the repolarization of the ventricle, okay, and that would be the start. So you can see here the ventricle is starting to repolarize, and so that would be the start of the T wave. And then when the T wave finishes, the ventricle has completely repolarized and, in essence, also relaxed. Okay. Now, sometimes you kind of have some blips or kind of strange things that might be going on. So here you can see P wave, the QRS T wave. Sorry, T wave. P wave, QRS T. But the T is kind of missing and the QRS is elongated. It's wide. So instead of being sharp, it's wide. So that would indicate that it's taking longer because this graph is seconds is on the x-axis. Okay, so it's taking, or in this case milliseconds, it's taking longer than a normal EKG to go through the QRS. Okay, so if you see this wide QRS, it tells you there's something wrong with the ventricle or there's something wrong with controlling the ventricle. Normally when we see this, it indicates that there's AV bundle damage. Okay? And so we'll go through quite a few of these EKGs and we'll look at them and try to decipher, well, what does it mean when there's no P wave? If there's no P wave, maybe instead of the AV bundle damage or an AV issue, maybe it's an SA node is not firing correctly. Okay? If there's no T whatsoever, okay, then maybe it means that the ventricle is not repolarizing, okay, and maybe there's an issue with the myocardium and the ventricle. Okay, so we'll look through some of these and talk about some different issues that can occur. All right, so how does pressure and volume change during a cardiac cycle? Okay, so pressure is what's really controlling a lot of the valves. Okay, so the papillary muscles that you saw attached to the bicuspid valves, so the cuspid valves, the bicuspid and the tricuspid valve, so the mitral valve and the tricuspid valve, that's controlled by the electrical impulse that's generated from the heart. Okay? But the semilunar valves, they're more controlled by the pressure that's occurring in the ventricles. And so early on, prior to stimulation, what you're going to have is the atria is going to fill. Okay, the atrial is filled by vein blood that's lacking oxygen, deoxygenated, or not rich in oxygen, making it back to the right atrium via veins. So the inferior superior vena cava, dumping it back into the heart. Okay, so that right atria is going to fill with blood. It fills with blood, okay? But the tricuspid valve is open. Okay? And when that fills, that tricuspid valve is open, and it allows through just passive leakage, it allows the blood to move from the atrium into the ventricle. Okay, and that's passive. So about 70-ish%, depending on the individual, about 70% of the blood that moves into the atria is just moving into the ventricle passively. Okay, there's no force, there's no muscle contraction. Then you have a muscle contraction, a systolic, atrial systolic contraction, and a ventricular distal relaxation. Okay, so contraction, the SA node is now sent the impulse. Okay, now those muscles contract. That forces the blood from the right atria and the left atria, it's both at the same time. Okay, it forces the blood out of the atrium into the ventricles. Okay? That's where kind of the 30% comes. So the remaining 30%, so this 70% is passive, the remaining 30% or so is pushed into the ventricles. Okay? The semilunar valves are closed at this time. Okay? So the blood is only going through the atrioventricular valves. Okay? And so it gets into the ventricle. That causes the pressure in the ventricle to increase. Okay? So when that pressure rises to a higher pressure than the atrial pressure, the AV valves are closed. Okay? When these AV valves are closed, then the only place for the blood to go is through the semilunar valves. Okay, so the atrial will relax. That increases the pressure in the ventricle side of the heart. Okay, and then once that atrium is relaxed, okay, the impulse is being sent to the ventricle part of the heart. Okay, the pressure continues to build, and then the semilunar valves, both the aortic and the pulmonary, open up, and the blood is forced out either the pulmonary, well, both pulmonary trunk and the aortic trunk or aorta. So let's look at that. Okay, so you got an initial SA node kind of contraction here. It's forcing that 30% from the right atria to the right ventricle, okay, through the tricuspid valve, okay, and then on the opposite side, the left atria is forcing the blood from the left atrium, okay, through a contraction to the left ventricle. The pressure is building up in both the left ventricle and the right ventricle. When it gets high enough, the atrial valves close, atrioventricular valves close, okay, and then the heart contracts, the ventricles contract and force the blood through the pulmonary and the aorta. Okay, and remember, these are controlled by semilunar valves. These valves are controlled by pressure in general. There's no muscle attached to these like the cuspid valves. There is muscle that's attached to them, so it's both muscle and pressure that's kind of determining the function of these guys.